Have you ever wondered how life on Earth became so diverse, from single-celled organisms to humans, elephants, and eagles? The answer lies in the process of evolution. Biological evolution is the genetic change in a population from one generation to the next. New allele, which are different forms of a gene, arise through mutations. When these new alleles enter the gene pool, they become subject to natural selection, and over time, the frequency of specific alleles may increase or decrease in the population. This change in allele frequency is what we call evolution, more specifically, microevolution in action. A species is a group of organisms that can interbreed naturally to produce fertile offspring. If you'd like to learn more, check out my video on the species concept. Members of a species that live in the same geographical area form a population. For example, the kokako on Tiritiri Island in New Zealand, lions in the Gir Forest in India, and coyotes in Yellowstone National Park are all examples of distinct populations. Each population has its own gene pool, the total collection of all allele in that population. The genetic diversity of a population depends on the variety of allele within its gene pool. It's important to remember that evolution acts on populations, not individuals. So, any change in the frequency of allele in a population's gene pool means that evolution is taking place. To calculate the frequency of an allele in a population, you divide the number of times that allele appears by the total number of alleles for that gene in the population. Several processes cause changes in allele frequencies and therefore drive evolution. These include mutation, natural selection, and gene flow, which is the movement of genes between populations. Another key concept that you need to understand here is evolutionary fitness, an organism's ability to survive and reproduce successfully. Fitter organisms are better adapted to their environment, and as a result, they are more likely to pass on their advantageous allele to the next generation. Now, let's talk briefly about mutations, the ultimate source of new genetic variation. A mutation is a sudden, random change in an organism's genetic material. It's important to remember that mutations do not occur because an organism needs them. They happen by chance. There are three main types of mutation: gene mutation, chromosomal mutation, and ploidy. 1. Gene mutation, a change in the base sequence of DNA within a single gene, often resulting in a new allele. 2. Chromosomal mutation, a structural change in a chromosome that can affect many genes. 3. Ploidy. Ploidy is their change in the number of complete sets of chromosomes, while the structure of individual chromosomes remains the same. Among these, gene mutations and ploidy changes play the most important roles in evolution. So we'll focus on those in this video and the upcoming episodes. Gene mutations often occur during DNA replication through base substitution, deletion, or insertion. While some mutations happen spontaneously, many are caused by external agents, known as mutagens. Mutagens include high-energy radiation, such as X-rays and gamma rays, and chemicals, such as benzene, formaldehyde, asbestos, and mustard gas. Certain viruses can also cause mutations, for example, the hepatitis B virus, which can lead to liver cancer. Fortunately, DNA has repair enzymes that constantly scan and fix most replication errors. However, when this repair system fails, mutations can persist, and sometimes these changes become the starting point for evolutionary change. Now, let us examine the different types of gene mutations. A substitution mutation occurs when one base in the DNA sequence is replaced by another. It changes only a single nucleotide, which may or may not alter the amino acid in the resulting protein. Substitution mutations can be of three major types. 1. Missense mutation. A missense mutation happens when just one base in the DNA changes, causing one amino acid in a protein to be replaced by another. This can slightly change the protein, or completely alter its function. For example, sickle cell anemia is caused by a single missense mutation in the hemoglobin gene. 2. Same-sense, or silent mutation. Same-sense, or silent mutation also changes one base, but the new codon still codes for the same amino acid. Because of the genetic code's redundancy, the protein remains unchanged, and there's usually no effect at all. 3. Nonsense mutation. A nonsense mutation is more serious. Here, a base change turns an amino acid codon into a stop codon. This causes the protein to stop being built too early, producing a short, incomplete, and usually non-functional protein. Now, let us examine insertion and deletion mutations. Insertion mutations add extra bases to the DNA, while deletion mutations remove them. If the number of bases added or deleted isn't a multiple of 3, it causes a frameshift. From that point onward, every amino acid changes, often resulting in a completely non-functional protein. Thanks for watching this first episode in our series on the process of evolution. In this episode, we explored gene mutations as a major source of genetic variation. In the next episode, we'll take a closer look at ploidy, another important type of mutation. Until then, happy learning.